A rat should freeze at the smell of a cat. That fear is wired in before birth, but infected rats walk straight toward it instead. Stanford researchers went looking for why, expecting a behavior quirk, not a mechanism. What they found wasn't in the rat's brain at all. It was inside the parasite itself, something no one expected a single-celled organism to be capable of making. The parasite is called Toxoplasma gondii, a single-celled organism that can only reproduce sexually inside the gut of a cat. To get there, it needs a host animal like a rat or mouse to end up inside a cat's stomach. So the parasite does something remarkable. It changes the rodent's brain to make that outcome more likely. Researchers Ajay Vyas and Robert Sapolsky at Stanford University showed in a study published in the Proceedings of the National Academy of Sciences in 2007 that infected rats lose their normal fear of cat urine and become mildly drawn to it instead. A follow-up study by Patrick House, working in Sapolsky's lab, and published in PLOS One in 2011, went further. Using brain scans, the team found that infected male rats showed increased activity in the brain circuits tied to sexual attraction when exposed to cat odor, on top of reduced activity in the fear circuits. The parasite was not just muting fear. It was rerouting the signal into something that looked like attraction. This effect only shows up around cat smell specifically. Infected rats still avoid open spaces normally and still react normally to other threats, like the sound cues used in lab experiments to signal a mild electric shock. The manipulation is narrow, aimed at one single behavior that helps the parasite complete its life cycle, not a general breakdown of the animal's brain. Other researchers have gone looking for a mechanism behind this precision, and they found something unexpected inside the parasite itself. Studies published in PLOS One and other journals found that toxoplasma carries its own copy of a gene for tyrosine hydroxylase, the enzyme responsible for producing dopamine, a chemical messenger tied to motivation and reward in the brain. Infected brain tissue shows elevated dopamine activity directly inside the dormant cysts the parasite forms, suggesting the organism may be manufacturing its own dose of a brain chemical to help steer its host's behavior. Some studies have even shown that drugs used to treat dopamine-related psychiatric conditions in humans can block the behavior change in infected rodents entirely. That precision is what makes this parasite so unsettling to study. It is not causing random brain damage. It is targeting one behavior with something close to surgical accuracy, using tools borrowed from the very chemistry that shapes emotion and motivation. And the strangest part of this story has nothing to do with rats at all, because the same parasite spends time inside human brains too. According to the Centers for Disease Control and Prevention, roughly 11% of Americans age 6 and older have been infected with Toxoplasma gondii at some point, and the agency estimates more than 40 million people in the United States currently carry the parasite. People usually get it from handling cat litter, eating undercooked meat, drinking unpasteurized milk, or touching contaminated soil while gardening. Once inside a person, the parasite forms small dormant cysts in muscle tissue and in the brain, where it can stay for the rest of that person's life, occasionally rupturing and releasing more parasites into the bloodstream. For almost everyone with a healthy immune system, this causes no noticeable illness at all. The CDC and multiple published studies estimate that around 90% of infections in people with normal immune systems produce no symptoms whatsoever, or symptoms so mild they get mistaken for a passing flu. The parasite is simply there, quietly encysted, doing nothing that a person would ever notice on their own. The exceptions matter. Pregnant women who get infected for the first time during pregnancy can pass the parasite to the fetus, and the CDC estimates this happens in several hundred to a few thousand pregnancies in the United States each year, sometimes leading to serious eye or brain damage in the child. People with weakened immune systems, including those with HIV or AIDS, face a much higher risk of the dormant cysts reactivating into a dangerous brain infection called toxoplasmic encephalitis, which for years was one of the most common severe neurological complications in AIDS patients, before modern antiretroviral treatment became widely available. For everyone else, the harder question researchers have spent years chasing is whether a parasite capable of rewiring fear circuits in a rodent brain does anything measurable in a human one. Several research groups, including scientists associated with the Stanley Medical Research Institute, have published studies linking toxoplasma antibodies to subtle changes in reaction time, memory, and mood, and some have explored a possible connection to schizophrenia risk. Based on the same dopamine pathway the parasite appears to manipulate in animals. None of this research claims the parasite controls human behavior the way it controls a rodent's. The findings are correlational, meaning they show a statistical link, not proof that the parasite is the cause. Scientists studying this are careful to say the picture is still incomplete. Toxoplasma is not the only organism turning up in American backyards capable of reaching a human brain. Unlike toxoplasma, the next parasite does not need precision or a borrowed brain chemical. It just needs a child willing to put dirt in their mouth. Balisascaris procyon, commonly called the raccoon roundworm, lives as an adult worm inside the intestines of raccoons and produces eggs that get passed in raccoon feces. Raccoons often use the same spot repeatedly as a bathroom, called a latrine, usually at the base of a tree, on a woodpile, or on a flat rooftop. And a single raccoon latrine can contain millions of these eggs. The eggs can survive in soil for years, resistant to freezing temperatures and most common household disinfectants, which is part of why the CDC lists this as an emerging concern rather than a fading one. Humans are not a normal host for this worm. When a person, usually a young child, accidentally swallows the eggs from contaminated soil, sandboxes, or wood piles, the eggs hatch into larvae inside the intestine. Unlike in a raccoon, the larvae in a human do not stay put. They migrate through the body's tissue in a process doctors call larva migrans, and when they reach the brain, they cause a severe condition called neural larva migrans. And when they reach the eye, they can cause permanent vision loss. The first confirmed human case was reported in 1984, involving a 10-month-old infant who died from the infection, with autopsy findings showing larvae embedded throughout brain tissue, most heavily in the brain itself. Since then, the CDC and published medical journals have documented several dozen confirmed human cases in the United States, concentrated in states with dense raccoon populations, including California, New York, Minnesota, and Washington. And researchers have also tracked the parasite's geographic spread into new states like Florida in recent decades as raccoon populations near human homes have grown. Most diagnosed cases have occurred in young children under 2 years old, though the CDC notes that older patients are being diagnosed more often as awareness of the disease grows. At least 5 of the documented cases have been fatal, and many survivors are left with permanent neurological damage, including seizures and loss of motor control. Before we move on, if you're enjoying this analysis, please consider subscribing. It helps the channel tremendously. Doctors describe this infection as rare, but almost certainly underreported, since testing for it is difficult, and many mild or borderline cases likely go undiagnosed entirely. That gap between how often it may actually happen and how often it gets caught is exactly what makes the next part of this story so important. Rabies is not caused by a parasite in the biological sense, but it belongs in this story because of what it does to an infected brain. The rabies virus travels along nerves until it reaches the central nervous system, where it triggers dramatic behavioral changes before killing its host in nearly every case once symptoms begin. According to the CDC, more than 90% of rabies cases reported in the United States occur in wildlife, with raccoons, skunks, bats, and foxes serving as the main reservoirs for the virus, each carrying its own regional strain. Most people picture a rabid animal as aggressive and foaming at the mouth. And that version, called furious rabies, does happen. A 2011 study of confirmed rabies cases in Massachusetts, published in the journal Animals, found that aggression was a strong statistical predictor of infection in wild animals, with gray foxes, red foxes, and coyotes showing the strongest link between aggressive behavior and a positive rabies test. But rabies has a second, quieter form, sometimes called dumb rabies. In an interview with NBC News in 2025, CDC rabies team lead, Dr. Ryan Wallace described a docile strain where infected animals become unusually friendly and lose their normal fear of people entirely. Wallace described a case where a family petted and fed a sick raccoon that approached their front porch, not realizing the unusual friendliness was itself a symptom of the disease, until the animal died. The CDC reported in 2025 that it was tracking 15 separate likely rabies outbreaks across the country, spanning states from New York to Oregon, with 6 human deaths in the previous 12 months, the highest yearly total in years. Around 100,000 Americans receive preventive rabies treatment, called post-exposure prophylaxis, every year after a possible exposure, a number that reflects both genuine risk and the seriousness with which doctors treat any bite or scratch from an unfamiliar wild animal. Rabies is almost always fatal once symptoms begin, which is exactly why public health officials treat any unusually bold or unusually friendly wild animal as a potential warning sign rather than a curiosity worth approaching. What connects a friendly raccoon, a fearless rat, and a child who touched contaminated dirt is not that any of them were unlucky in some random way. It is that all three involve an organism reaching into a brain and changing what that brain does. And it is exactly that overlap that makes diagnosing these cases in a hospital so difficult. Neural larva migrans from Balis ascaris and toxoplasmosis in the brain often show up on an MRI as inflammation in the brain's white matter, a pattern that looks similar to several other infections and autoimmune conditions doctors see far more often. Doctors typically diagnose these conditions by testing spinal fluid for elevated white blood cells, called eosinophils, a sign the body is fighting a parasite, then narrowing down the cause through blood antibody tests and a detailed history of the patient's exposure to animals, soil, or undercooked meat. A case published in a 2022 medical journal described a 19-month-old boy who arrived at a hospital with unsteady walking and was eventually diagnosed with Baylis ascaris, meningoencephalitis, only after doctors consulted parasitology experts at the CDC directly, since no simple, widely available test exists for the infection. Cases in older patients get missed even more easily, since doctors are trained to expect this kind of infection in toddlers, not adults. A 2016 case report published in a CDC journal described a 63-year-old man who developed severe neurocognitive decline before doctors correctly linked his symptoms to raccoon roundworm exposure near his home, ultimately confirming the diagnosis through antibody testing after ruling out more common causes. This is not a story about hospitals failing patients. Reported case counts likely undercount how often these infections happen, exactly because the symptoms overlap so heavily with other, more familiar illnesses that doctors are trained to look for first. And that gap between real-world exposure and confirmed diagnosis is the same gap that connects every organism in this story back to one simple fact about how nature actually works. None of this is science fiction. Toxoplasma gondii really does rewire fear circuits in rodent brains using its own borrowed dopamine chemistry, and it really is living inside an estimated 40 million people in the United States right now, almost always without symptoms. Baylis ascaris prussianus really has caused fatal brain infections in American children after contact with raccoon droppings, and cases are still being identified in new states as raccoon populations near human homes continue to grow. Rabies really does alter animal behavior in ways trained wildlife officials still find surprising, sometimes making an animal more dangerous through aggression, and sometimes more dangerous through unnatural friendliness that invites the exact kind of contact people should avoid. What ties these together is not a hidden conspiracy or a monster waiting in the woods. It is a simple biological fact that a handful of organisms found across American backyards, parks, and farmland have evolved ways to influence the nervous systems of the animals they infect, and humans are not always excluded from that reach. Researchers at the CDC, at Stanford, and at hospitals across the country continue documenting these cases because understanding exactly how far that influence goes is still, after decades of study, an open question, and every new case adds one more piece to an answer nobody has fully worked out yet. If you enjoyed this video, make sure to subscribe, and I'll see you in the next one.